Hello everyone, welcome back to my channel. My name is LEE ZHI XUAN. So this video is the free class video for STPM Biology Semester 2, Chapter 13, Immunity. So this will be the last two chapter in this semester, which is semester 2. So this video is special to design for those who are learning the STPM Biology Semester 2 lah. So let us to see, based on Majlis Peperiksaan Malaysia MPM, what you would need to learn in this chapter. So in this chapter, it is about the human immunity. So in this chapter, we only learn about human reaction. So in this chapter is the one of the chapter that you don't need to learn about the plants. The plants immune system, animal immune system, that one you don't need to learn. You only learn about human only. So this chapter can be said that it is totally new chapter for you that you never learn in SPM. So although you got learn immunity part, immune system in SPM, but this immune system is more deeper compared to SPM. Okay, so there are 4 big topics that you need to learn in this chapter lah. So first of all is about immune system. What is the human immune system looks like? And then the second part is the most important in this chapter, which is the development of immune system. How our body fight against the pathogens. Another one is concept self and non-self. And then the final one is immune disorder, meaning that when your immune system is functioning have problem, what is the effect? So they have the disease lah. Some of the disease that you need to learn lah. Okay, so these are 4 big topics that you need to learn in this chapter.
In this lesson, I'll be using these figures to guide our explanation. If you've already purchased my notes, you'll find the full, detailed explanations written out there to follow along. If you'd like to get your own copy of these notes, feel free to contact me directly.
First, we will look about the basic overview of immunity. So what is definition of immunity? So I think everyone already know. So immunity actually is defined as the resistance to the disease, specifically is infection disease, meaning that is how our body fight against the disease. In any type of disease, including infection disease. Not only infection disease, including also the cancer, pathogen, including cancer cell also is one type of the immunity function. Okay, so this is actually what I need to recap to you guys. In your SPM, the immune system that you learn is only we call as first line and second line. It is an innate immune response that you already learned in SPM. If you still remember in your SPM, you know that you got three line defense system, right? First line, second line, and third line. And then first line and second line is the basic one, and then the third line is in your SPM, you say you will use antibody. Antibody will be used in the third line defense. And then first line is the very basic one, is your skin. It is a first barrier, physical barrier. And then second line, you will use the white blood cell, including neutrophil, monocyte, all that, phagocytosis. That one is that you already learned in SPM. This is what we call as in scientific term, we call as innate immune response. So in STPM, we will not learn this. The first line and second line that you already learned in SPM, we will not learn it. We in STPM, we focusing on third line, which in scientific term is we call as adaptive immune response here. This is a third line defense. So what is the differences between them? Normally, first line and second line is just the initial protection, meaning that they are not specific. Is there any type of pathogen, any type of bacteria, they will fight against the infection. However, in third line, it is more developed more slowly, and it is highly specific. So meaning that when the bacteria enter inside your body, in your blood, enter to your blood, the first line and second line will be take action first, especially second line. Your phagocytosis process, your macrophage, your neutrophil, your monocyte, will undergo phagocytosis to in the second line first. But third line still not yet happen. So how the third line be happen? This is what you need to learn. And when third line happen is when the second line is failed. Meaning that your neutrophil, monocyte, macrophage, all that still cannot destroy the pathogens. Then they will start to third line defense system. So normally in third line is, you can say, it's more than dangerous to you lah. So normally if the person go through the third line, normally you will get fever lah. Of course you will get fever because when during the development of the immune system, your bacteria still can infect other type of cells first. So it is like a time running lah. Either your immune system is developed more quickly, or your bacteria or the pathogens infect the cells more quickly. This is like the time racing. Either is your immune system develop quickly, or your infection of the pathogen develop quickly. So this is a time. So first of all, of course, you need to learn basic lymphatic system that you already learned in chapter 9 transport. But in chapter 9 transport is about the flow of the lymph fluid, all that only. How the lymph interstitial fluid, your lymph is formed, and how the lymph recirculate back to the blood circulatory system from the lymphatic system. But in this chapter, we will more focus on the lymphoid organs. So this is the diagram that you will learn in the need to learn about the lymphoid organ. So including lymph nodes lymph nodes is everyone know. So thymus, spleen, bone marrow also one type of the lymphoid organ. Tonsil. So normally you just need to know the most important thing is lymph node, thymus, spleen, and bone marrow. These four are very, very important. Important, especially lymph node. So first of all, we will see bone marrow. Okay, so bone marrow in basically we are considered as primary lymphoid organ. Okay, so before I proceed to the bone marrow, so you can see the green color one is we call as primary lymphoid organ. The red color one here is secondary lymphoid organ. Lymphoid organ. So in bone marrow, so this is one type of the anatomic structure of the bone marrow. So bone marrow function, I think everyone already know, is play the role in the synthesis of the white blood cell, and also red blood cell. All the development, initial maturation, initial development of the blood cell is from bone marrow. So that's why that when the bone marrow have problem, then your blood cells will have problem also. Because it is the industry to develop, to produce the blood cells, because it containing hematopoietic stem cell. When you see the stem cell terms, meaning that it can be reproduced and divide uh, unlimited. No limited one, unlimited one. So it can produce many and many blood cells unlimited until you die. So this is the function of the bone marrow. Lymphocyte is play the role in the adaptive immune response that you already know from the last chapter, right? So this is the bone marrow structure. So you can see there are artery, veins, all that. Okay, so like you can see, you got one cell we call as osteoblast. So normally this is the osteoblast. So osteoblast, I think, is the bone cell, right? That you already learned in also last semester. Bone tissue. Connective tissue, one type of the example of the connective tissue is bone, right? So osteoblast is the basic cell of the bone. So it also contained in the bone marrow as well. So it generate the bone cell and also control the differentiation of the homeopoietic stem cells. And then there are also another cell. We can see another cell is the endothelial cell. Endothelial cell, we also another one is endothelial cell. Endothelial cell at the surrounding of the bone marrow. So this is a line on the blood vessel, also regulate the hematopoietic stem cell differentiation. Another one you can see also, another one is reticular cells. So this is actually to send the process and connecting cell to the bone and blood vessels. And then also there are also neuron. Of course, there are neuron cell that play the role in the release of the homeopoietic stem cell from the bone marrow. So this is all about the bone marrow. So yeah, you just need to know the major function. The major function is containing HSCs, and then can develop produce B lymphocyte, not only lymphocyte, all the blood cell. And then the development and maturation is happening for the B cells. Okay, so next one is thymus. Okay, so this is the structure of the thymus. Thymus here. Normally, thymus is located in your above the heart. This is the heart, right? This is your heart, and the thymus is located above your heart only. So it is also another type of the primary lymphoid organ, play the major role for maturation of B cell. So bone marrow is play the major role for maturation of B cell. B cell. You will still remember lymphocyte containing B cell and T cell, right? That one I will explain more detail. What is B cell? What is T cell? Okay, that one you already know, right? Lymphocyte containing B cell and T cell. So B cell is that mature at bone marrow, but B cell and T cell, both of them are produced, are synthesized from bone marrow. After that, your T cell will go to the thymus for maturation. But B cell still maintain at your bone marrow. It will not go anywhere. You just stay at bone marrow for maturation. But T cell will go through the thymus for maturation. So this is a key function of your thymus. 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 So that thymus is specialized for the environment for immature T cell, T cell to generate the unique receptor we call as T cell receptor on the surface of the cell. So this is a thymus major function. Okay. So another one, this one lymph node is the most everyone already know lah. I think I think in SPM also you already know. So in SPM, you know that lymph node is play the major function in the infection. It containing all the lymphocyte and play the function in the infection. Yeah, your all the majority of your third line defense system is happening in your lymph node. Majority. So it is the most specialized secondary lymphoid organ that are encapsulated bean-shaped structure that packed with many white blood cells, including your lymphocyte, macrophage, and also another white blood cell is dendritic cell. So majority are happening at here. So you can see here, major role is to regulate the adaptive immune response, which is the immune response, which is the third line defense system, to activate the B cell and T cell. So for now, maybe you are not very blur what I'm talking about. So all the place here is all the development of the B cell and T cell, and also the development of the immune response, that in the second big topic, development of the immune system, is happening at here. Lymph node. Majority is happening at here. So it provide the environment to encounter the relationship between the antigen and also the lymphocyte. So all the antigen from your blood, in the blood, will be go through your lymphatic system, and then antigen will come out, will go to the lymph node. Of course, your lymphocyte also will go to the lymph node. So it provide the environment that lymphocyte can kill your antigen. Just simply like that. Simply like that. But the process for how the lymphocyte kill the antigen is very complicated process that you will learn. That I will talk about that lah. But for now, you just remember that lymph node is provide the environment that lymphocyte can kill the antigen. That's all. That's all. So in the lymph node, you can imagine it's like the kidney lah. The shape is look like the kidney. So it also containing the cortex region and the medulla region. Cortex is the outer part, and also the medulla region is the inner part. So normally your cortex region is containing the B cell. B cell, and then another region is the at the center one, between the cortex and the medulla, we call as paracortex. This region is more containing T cell. T cell. And then the medulla is the innermost layer, which is the place where the lymphocyte can in and out. Like it containing the blood vessel lah. Containing blood vessel, which that the lymphocyte, white blood cell can move in and out. Because you need to know that in lymphatic system during the formation of interstitial fluid, white blood cell cannot pass through one. Ha. Your limb and interstitial fluid does not containing any white blood cell, right? So how the white blood cell can enter lymph node? So surrounding the lymph node in the inner layer part, they're containing the blood vessel, blood capillaries. So these blood capillaries allow the lymphocyte go to the lymph node so that it can diffuse out from the blood capillary into the lymph node lah. So this is how the your white blood cell can enter into the lymphatic system. Lymphatic system. So this is the site where the lymphocyte in and out lah, from the lymph node. Okay, and then after that, of course, there are also the pathway where your antigen can go through go into the lymph node by the afferent lymphatic vessel lah. Lymphatic vessel. So yeah, this is what we call as afferent lymphatic vessel lah. And then there are also the efferent one. So efferent is the go out lah. Afferent is incoming, and then the efferent is outgoing. So all the antigen were coming from here, from the afferent lymphatic vessel. So once the antigen is already entered at here, then all the development of the immune system, adaptive immune response will be happen at here. Later you will learn about that. About that. Okay, so that's all about the lymph node. So this is a lymph node structure that you need to know lah. You need to know. Okay. Then another one, secondary spleen, lymphoid organ is we call as spleen. Okay. So this is the structure of the spleen. So normally it is situated high in the left hand side of the abdominal cavity, which is the large ovoid secondary lymphoid organ. So look like that. So when you see the more majority part, you can see spleen is located at here. Left hand side, right? He is human, so it located at your left hand side. Left hand side at your abdominal cavity. Normally near the lung, near the lung part. This is what you call as spleen. And only one. Each human only containing one spleen. But lymph node have many. And you can see all these, you can see many like the loops structure. One, these all are called as lymph node. But spleen only have one. Okay, so what is the major function of your spleen? So the play major role in mounting immune response to antigen in the bloodstream. So this is very important. Differences between lymph node and spleen. Lymph node, normally all the antigen is coming from the infection site. It is not coming from the blood one. It is coming from the infection site of your infection site of the organ. Any organ maybe your heart is infected. So this antigen will come. Heart is infected. So this antigen is coming from heart. One. Okay. So spleen is more focusing on bloodstream, meaning that when your blood containing any antigen, then this spleen play the major role to activate it. So it also play major role in the development of the immune response, adaptive immune response, but the antigen sources is different. The antigen sources for the spleen is from the blood. So when the lymph node are specialized for encounter between lymphocyte and antigen drained from the local tissue. The spleen are specialized in filtering blood and trapping blood-borne antigen. So these are two key difference. Spleen play the role in filter the blood. So you can see this spleen structure like this. This is a spleen artery, right? This is a blood vessel that's surrounding the spleen. When it come through, and then it will go through the spleen, surrounding the spleen. And then at here, the spleen will filter up the blood in the artery to see whether there are antigen or not. If there are antigen, then it will filter out and do the adaptive immune response. So here, spleen is particularly important in response to systemic infection. So any antigen in the blood are carried into the spleen through the artery and out by the vein. So like this in, and then it will out by the vein. So you can see here, all this blue color is vein, spleen vein. So spleen is surrounded by a capsule from which a number of projections known as tubular extend, providing the structural support. Not sure. These are. So this is all these structures we call as spleen. So normally in spleen we containing the two part, which is red pulp and white pulp here. Red pulp and white pulp. Okay, what are they? Okay, so red pulp normally is consists of a network of sinusoid populated by the red blood cell, macrophage, and also the lymphocyte, like this. And then the white pulp is at here. White bulb is at the, you can see the brown color one, containing follicle, containing marginal jaw, containing PALS. PALS. So normally it's a bunch, surround the bunch of the splenic artery. Red bulb is more on the vein one. White bulb is at the artery part. So it is play the major function to as the first line lah, for the filtering the blood antigen from the blood. Okay, so this is what we call as spleen lah. Okay, there are also other type of the lymphoid organ which we call as mucosal associated lymphoid tissue, including tonsil, appendix, and prayer patches. So this is actually a group of the lymphoid tissue that play the defense vulnerable mucosal membrane surface act as the major site or the entry for most pathogen. So normally these tissue are located in the mucous membrane. The mucous membrane. So including example is like intestine. One of the major example is in the intestine that containing mucus and including our nose that any membrane that can able to secrete mucus is containing this all the three lymphoid organ. So yeah, so actually this one is just additional information for you lah. You don't need, I think it will not become out. I just add into my notes. But the most important thing is here: spleen, and also thymus, lymph node, bone marrow. These four organs that you should know lah. But this one you can, you just can go through it. Okay. Next is about the development of B cell and T cell. Okay, so in third line defense system, there are three major white blood cells that you must know. First one is macrophage. Yeah, macrophage. So later you will learn what is the function of the macrophage. Okay. The next one is B cell and T cell. Okay, so this one, development of B cell and T cell actually is, I can say actually in real situation, it is very complicated development process. I think it should not be in your syllabus. Yeah, it should not be in your syllabus. However, in the MPM syllabus, they need you to describe the development of B cell and T cell. Because they already mentioned want you to describe. So yeah, even in the old syllabus, they still also want you to describe. But in real situation, even you can see the development of B cell and T cell actually is highly complicated. So and Fasil also never come out this kind of question yet. But since it in the syllabus, so yeah, I need to teach. I need to include it lah. So yeah, so it's very complicated. I hope I can use the easy words to get you understand. So back to the normal world. In the blood cell, not only lymphocyte, including all the blood cell, including red blood cell or all the white blood cell, monocyte, neutrophil, eosinophil, basophil, platelet, all that is produced in bone marrow, as I mentioned. So because bone marrow containing hematopoietic stem cell, it is can able to renew and reproduce divide automatically one. So it can, along your life, you can produce many and many blood cell that require survive in your life. So in development of B cell and T cell, after that, T cell is naturally is coming from the lymphoid progenitor cell. So after your homostotic stem cell will differentiate, either is differentiate into myeloid or lymphoid. And that for myeloid, later it will develop into dendritic cell, including monocyte, neutrophil, all other type of the other than lymphocyte, all the cell is include from the initially is from the myeloid progenitor cell, including all this. You can see here, but they will also differentiate into lymphoid progenitor cell. So in lymphoid progenitor cell, it will develop three types of major cell, including natural killer cell. So this one is not in your syllabus. You don't need to learn. And also dendritic cell, also not in your syllabus. You don't need to learn. So the major part is here. T cell and B cell. Okay. T cell and T cell. It will develop into T cell and B cell. And then in T cell, there were two major exact types that you need to know. Not only these two lah. Not only these two. In real world, not only these two. But in your syllabus, you just only learn about these two only. Helper T cell and cytotoxic T cell. So some of the books say T4 and T8. T4 cell and T8 cell. And also some of the books say CD4 and CD8. Different type of the reference book, they have different type of the uh short form abbreviation. TH and TC is that this one is using lah. So TH is helper T cell, TC is cytotoxic T cell. There are also another book they use T4 and T8. T4 is helper T cell, T8 is cytotoxic T cell. Another book use CD4 or CD8. So helper T cell have many abbreviations. TH, T4, CD4. These three. So if you see these three in your question, TH, T4, CD4, all these are considered as helper T cell. For cytotoxic T cell, there were TC, T8, and CD8. CD8. If you see these three terms in your question, then it will be in cytotoxic T cell. So for me, my recommendation is when you want to write in the exam, especially in essay, you should write full name first. Helper T cell, then bracket. Bracket, either you want use what? You can use TH or T4. I recommend you use T4 and TH. I not recommend you use CD4 because if you use CD4, you will confuse. Because CD4 is more scientific. It's more scientific. If you not mastering well yet, because for me I already know because I got learn more deeper in my university, in the immune system. So I already know CD4, CD8. But if you don't know yet, please use TH or T4 for short form. So for cytotoxicity cell is TC or T8. T8. So this one is T cell. But for B cell, just simple, only one. B cell only one. B cell don't need you. There are no many type of B cell. B cell only one. So this is a majority of the uh all about the development of the lymphocyte and how the lymphocyte is formed. So they are all from initial is coming from lymphoid progenitor, and this lymphoid progenitor is coming from homopoietic stem cell. So okay. Next, we go through the development of T cell. Okay, so I hope in this video I will just go through only. I will not explain all this in detail because in real examination I don't think it will come out because it's very complicated. I know it's very complicated. Because if you want to develop a mature T cell in the thymus, you know, right? These are all the process that you need to know here. But I try to use the simple word, let you understand what is happening here. Okay, so these two part I already mentioned. Homeostatic stem cell. After that, it will differentiate into the lymphoid progenitor. After that, it will migrate into the thymus. You need to know thymus is the place for maturation of T cell. Bone marrow only produce T cell only. They just produce lymphoid progenitor only. After that, this lymphoid progenitor will go to the thymus for maturation for spatial for T cell. Okay, so this is all the process. So the most important thing is here, positive selection and negative selection. Okay, so this part actually is more complicated before you learn about the immune system first, and also the concept self and non-self. So they are all linked together. So before I proceed to here. So I don't want to talk much about this. Later I will come back. After you already know about the concept cell and concept non-cell and also the development of the immune system very well, then I come back. Then you come back. So it's more easy to let you know lah. So I assume you already watched the later part, then you come back here, Okay. So what is happening here? Okay. So as I mentioned, T cell containing two major types, CD4 and CD8, T4 and T8, T4 and T8. Okay. How your timers know? I want to develop this T4. I want to develop this to T8. How? So they have mechanism. So all these mechanisms. So about here, you don't need to know. You don't need to know. You just want to know here is to develop the T cell receptor. So in T cell, they have receptor we call as CD4 and CD8. So that's why that some of the book use T4 and T8. T4 and T8 because this 4 and 8 is coming from the receptor name. The receptor name is we call as CD4 and CD8. CD4 and CD8. Okay, CD4 and CD8. So once they already express the receptor, it will CD4 receptor and CD8 receptor, they will undergo positive and negative selection. Okay, so what is the meaning by the selection? The selection. So meaning that in the development, your immature T cell actually is containing both CD4 and CD8 one. They're containing both one, CD4 receptor and CD8 receptor. So they need required to undergo the positive and negative selection depending on the signal they receive when they encounter the self MHC or self peptide combination with their T cell receptor. Okay, if you already know the concept self and non-self, in natural, our T cell will bind to the antigen which is non-self only. If this is the self antigen, the T cell will not trigger any adaptive immune response one. Ah, yeah. So for now, negative selection meaning that they need to bind it. Depends what kind of signal they receive lah. So that kind of signal that you don't need to know because that kind of signal is very highly complicated one. Complicated one. Once they receive the signal, they will encounter self MHC or self peptide combination. So your receptor will bind to the peptide immediately which are positively selected and made sure to single CD4 or CD8. So once they already receive the signal, they will only express one type of the receptor only lah, CD4 and CD8 only. So in the beginning, they are double positive. Okay. Once they already receive the signal, then they will remove either one of the receptor. So either they remove CD4 or they remove CD8 lah. CD8. Okay. So this is what we call as the positive selection. After that, they will go through the negative selection, which they will well developed with the CD4 protein and CD8 protein. Protein. They will undergo the negative selection to mature your CD4 and CD8. So this negative selection I can say in general terms, is just us able to select the differentiate self antigen and non-self antigen. Both of them selection, positive selection is to choose. Easy. Choose either CD8 or CD4. Want to maintain CD4 or CD8 only. Negative selection is to differentiate self and non-self. If if the this cell CD8 cell is able to trigger the reaction for the for the cell, then it will eliminate it. We don't want that. We don't want that because we don't want your white blood cell, your T cell can attack your body one. You need to know in our whole body, all of your cell containing antigen one. Either these differences is itself or non-self. You need to know antigen, and the terms antigen is not only coming from the pathogen. Even our healthy cell also containing antigen one. So one of the major example is blood cell, ABO blood type. You know that ABO blood type containing antibody A, also antigen A. So that antigen, you think is it bad? No, it is not bad. It is a self antigen. So your CD8, CD4 should not bind to that antigen. If they bind to that antigen, meaning that your you will have the we call as immune disorder. So this is what we say as development of the T cell. So your T cell that express CD4 is helper T cell, that express CD8 is cytotoxic T cell. So this I already try to use my easy word to to explain the development of the T cell lah. So if you are interested, maybe lah, I don't know, if you are interested, want to know about more detail about the development of T cell, maybe I will create another video, special video lah for General it, but I need to see your interest, want to know or not. If you don't want to know, then yeah, I just try to use the easy word to explain to you lah. Okay. Okay, next is B cell. Okay, so this is a general view of the development of the B cell. Okay, so in B cell also containing stem cell, right? Homeostatic stem cell, and the maturation of the B cell is happening at the bone marrow. It is happening at the bone marrow. Okay, so B cell, the special case for B cell is it able to produce antibody. So antibody actually is produced from the B cell. So this is a overview of the maturation. So your maturation proceed through sequential step, which is characterized by the gene, by the antibody gene expression, expression. So it will either go homeostatic stem cell, it will develop into the pro-B, pre-B, immature B, finally it will go through the major B, major B. So at the pro-B and the pre-B stage, failure to express the functional antigen receptor will cause the death of the B cell. Meaning that if during this stage, the cell cannot express the receptor, before the B cell produce the antibody first, actually this is what called as B cell antigen. The B cell antigen actually is antibody. The differences between them is that B cell antigen still attached at the B cell. Once it release, it become antibody. These are the two difference. You can say that B cell antigen is equal to antibody. The differences is the location. B cell antigen still attached on the board and the B cell one still attached at there. But antibody is already released out. Once the B cell already release out the antigen, B cell antigen, then it become antibody. The terms the terms that you need to use. So in pro-B stage and pre-B stage is the stage that exposes the antigen. This is a process to express the antigen lah. So or including all the that you already know lah, transcription, translation, all that process. But this actually is very complicated lah. But I don't think you need to know lah. You need to know because if you want to learn about more detail about this, you need to know all the gene that found in the B cell. B cell. So yeah, because you are not the expert, you are just only pre-U student, you are not university student yet, so you don't need to learn more detail about this. But for those who want to go through the medical science, especially biomedical science, then you need to know the detail one, Okay. Okay, so as the differentiation proceed, your B cell is starting to express the cell receptor. receptor, once they already receive the signal from this receptor, it will induce the differentiation of developing of the B cell, and trigger the proliferation to trigger the maturation of the B cell. So this signal will allow the differentiation uh, of the B cell and through the early B cell stage to form the immature B cell that leave the bone marrow to complete the differentiation in the spleen. Yeah. Okay. So yeah, so that's all about the development of B cell. Yeah. Okay. So next we go through the antibody structure. Okay. So antibody structure. Okay, so you already know B cell can produce antibody. So now you need to know about the what is happening uh what is the type of the antibody that found in our body, in the human body. Okay, so hopefully you go understanding what is T cell and B cell. So this is a basic structure for B cell antigen. BD is B cell antigen because still attached on the B cell. If it attached on the B cell, we call this as B cell antigen. So you can see it look like the V shape, right? And it's V shape, something like V shape like this. Yeah, V shape. So in this, what you need to know is there are two polypeptide change, right? There are two polypeptide change, which we call as light chain and light chain and heavy chain here. The shorter part one is we call as light chain. The longer one that attached to the B cell is we call as the heavy chain. Okay, so this is the first structure. And then if you see at the end of the B cell, here, this part is we call as antigen binding site. This is a place where your antigen bind to it. All the antigen, any antigen, bind to it. But it is highly specific lah. But you need to know in adaptive immune response, third line defense system, all are highly specific. Only the specific antigen can bind to it. It like enzyme one. Enzyme. Only the antigen that show similar shape like this can bind to it. If the shape is not like this, then cannot bind. So this is antigen binding site. So that's why that in the at the antigen binding site, they have variable region and constant region here. So each heavy and light chain contain constant region and variable region. Variable region is at the outside one, at the outermost part. The inner part here is we call as constant region. So constant region is determine your class of the antibody. We have 5 type of the antibody, 5 classes of the antibody. Variable region here is to determine the antigen binding site specificity. So it actually determine the shape of the antigen binding site. So variable region consists of antigen binding site where the antibody bind to the specific epitope of the antigen. Okay, so the region are bonded by disulfide bridge. The constant region, you can see the antibody, well, the constant region in the light chain is bonded by disulfide bridge here. So so that's all about the basic structure of the antibody. Okay, so there are 5 class of the antibody that you need to know. So what you need to know is the function. Every class of the antibody have their different function. So this is a class IgG, IgM, IgA, IgE, and IgD. So the Ig terms is immunoglobulin. Immunoglobulin. The immunoglobulin is the scientific terms for antibody. Antibody is just a general term, but in scientific term, if you call it immunoglobulin, but in exam, you just write antibody IgG is already okay lah. Already okay. And then for this one, you don't need to know lah. Heavy chain. This is a heavy chain. They use the symbol. But that what you don't need to learn. You don't need to learn. You just want to know here. So you need to know, of course, their structure. So all the antibody structure is look like this. But this one is something that already there. The books are modified yet, but it is so still same, still look like this. So this antibody that already secreted from the release out from the B cell. So it will look like this. Okay, so what are their, you need to know their function. The first one is IgM. So this is the spatial antibody because it is a pentamer shape, which means that IgM is bonded by 5 antibodies. There are 5 antibodies bonded together to form IgM. So this is a pentamer structure. So all of that is bonded by disulfide, which you can see here. So these 5 all together to form pentamer structure is called as IgM. And it is the first antibody produced in the immune response. If you want to produce the first antibody, IgM is the first one. This is the first antibody produced. Also, the play major role including neutralization, agglutination, complement activation, all that that you will know. Okay. Next one is IgA. Okay, IgA here you can see. So IgA is dimer structure containing 2 antibody, right? So they are also bonded by the disulfide bridge. So IgA, the major is founding at the mucus region, the cell that can able to secrete mucus. It prevent all the pathogen attached to the epithelial surface. So normally it's the cell like intestine on the surface of the epithelium, including your intestine, your kidney tubule, all that is in here. Mucus. If you see the mucus epithelium, the keyword is IgA. So this is a free function of the IgA. Next one is IgD. Okay, IgD is the IgE, IgC. You can see they are monomer. Only one antibody, but the function are different. So IgD, the major function is to play the role of the B cell maturation. IgD play the function of the B cell maturation and activate B cell for the binding site. So it not play the role in the immune response one. IgD is play the cell in maturation of the B cell. So that's why you can see there are IgD at there. But so IgD does not play the role in the immune response. It's spatial. Okay, another one is IgE. Okay, IgE is the cell that normally play the role in allergic response. You see the allergic response, inflammation, and also the uh, allergic symptom and also inflammation is more focused on IgE and including the parasite. The parasite infection, inflammation, allergic response, all these are include for IgE. So another final one is IgG. Okay, so IgG, this antibody is the only antibody that can pass through the placenta. You still remember what I about the chapter reproduction one, chapter 11, if not, chapter 12, sorry, chapter 12, right, function of the placenta. So this one you can include in your function of placenta one. IgG is the antibody play the role pass through the placenta from mother to the fetus. So it play the role to protect the fetus one. Protect the fetus. So including all neutralization, agglutination, all that you already learned in SPM lah. All that is that it will be function, but the location is at the fetus. Protect the fetus, protect the baby. This is only the hormone can pass through the placenta. So IgM is the most general one. All you can say it is the first antibody reaction. Normally all the immune response can be react. IgA more focusing on the epithelial cell, mucus. You will see the mucus, epithelial, all are IgA. IgD does not play the role in immune response, play the role in B cell maturation, development of the B cell, maturation of the B cell. IgE more focusing on the allergy, inflammation, parasite infection is IgE. IgG is only for placenta. It is only the antibody can pass through the placenta, protect the fetus, protect the baby during pregnancy. So these are 5 class of antibody that you need to know. You need to know their basic structure, pentamer, dimer, another 3 monomer, and their basic function. That's all. Yeah. Okay. So next we will go through the key important thing in this chapter. Immunity development or development of immunity. This part is the most important in this chapter. Okay, so if I say you need to activate additive immune response, okay, I give you a case study like COVID-19 virus, coronavirus, COVID-19. When you infected with COVID-19, do you think first line and second line can be prevent your immune response or not? Like this. Yeah, you see here. Do you think first line and second line can destroy the virus or not? Coronavirus cannot. First line already failed because and virus already enter to your body, your skin, all your skin cannot prevent it. So you do the second line. Do you think all the neutrophil, basophil, eosinophil can phagocyte and then phagocyte the coronavirus? Cannot. So finally, only third line. Only your third line defense can kill the coronavirus. Okay. How the lymphocyte kill the coronavirus? How? So that is why that you need to learn here. You need to learn here. So before the B cell and T cell that already produced from the bone marrow and the thymus respectively, how they know, oh, this is coronavirus and I want to kill them. You can imagine that the B cell and T cell is the army, your shoulder. So what is the major role of your army? Army normally want to go to the war. You can imagine it's like a war, world war. Want to take part into the war. They require what? Require command. They require the command for the sergeant. If your sergeant does not give you the command, do you think the army want take part into the war or not? No, they require the command. Same as the B cell and T cell, they require the command first. So where is this signal come from? Where is this common command come from? Why? Where? This is what we call as activation of the additive immunity. You need to activate the B cell and T cell first. Let them know, okay, this is the coronavirus. I need you to kill them now. You need to tell them first. We call as activate. Because the B cell and T cell is the army. Or if you are not receive any knowledge, any information, and then I just sit there only. I will not doing anything. I just sit there. So they receive the information. So where is the information? So this is the information. Okay. That's why you need to know what is the another one is macrophage. Here. Macrophage. And what another term is we call as antigen presenting cell. So this is very important. Antigen presenting cell. So antigen presenting cell play the role to activate the immune system. If there are no antigen presenting cell, then all this cannot happen. No happen at all. Okay, so this is more general lah. So I'm more general part. So you can see here in antigen presenting cell, it was starting to activate your CD4 and CD8. So after that, you will go through all these reaction. So we will see how this reaction happen. Okay. So for the first one is macrophage. So normally first reaction is just like macrophage digest the bacteria through the phagocytosis, and then it will digest the bacteria. All the lysosome will feed with the phagosome to digest the bacteria. Okay, the final product is antigen. So this antigen would display on the surface of the macrophage. So the macrophage now is as the APC, which is an antigen presenting cell. So before the as an antigen presenting cell, antigen will bind to the MHC2 complex. So what is MHC? So MHC is a major histocompatibility complex, which we call as MHC. So it will bind to the MHC2, then it displays on the surface of the macrophage. And now your macrophage display this antigen. So imagine this using bacteria, right? If you my example is virus. You use a coronavirus. So here now is virus antigen. So the virus antigen is based on the macrophage already. Okay, so what is happening next? Okay, you already display. So what is happening next? So this actually is the signal. Signal to the helper T cell. So you still remember there are two types of cell, right? Helper T cell and cytotoxic T cell. So first one is helper T cell first. Helper T cell how come to the macrophage. So how it come? So once they already display the display the uh MHC2 antigen complex, we call as MHC2 antigen complex. So macrophage is act as the antigen presenting cell. Your ATC will release cytokine IL-1. Cytokine IL-1 to activate the helper T cell. This is very important. Cytokine IL-1. This is very, very important. Very important. Your so now your APC will display the antigen fragment, antigen complex with Cas2 MHC molecule on the cell surface. So your helper T cell here now will bind to this antigen fragment, and there are also the T cell receptor we call as CD4 will bind to the Cas2. Helper T cell containing antigen receptor. So this antigen receptor will bind to the antigen at that display by the APC. And there are also an accessory protein we call as CD4, like CD4 bind to the Cas2 MHC molecule. Bind to the Cas2 MHC molecule. So after that, your APC antigen presenting cell will release IL-1 to activate helper T cell. So this is the overall process. First, your macrophage, okay, coming from the blood, okay, dia jalan, okay, nampak coronavirus, okay, dia makan coronavirus ni. Okay, so that it will release antigen inside the macrophage, release antigen. So this antigen will bind to the MHC2 complex. And then it display the antigen. Now your APC, your macrophage is as APC lah, antigen presenting cell. Okay, once it already display, okay, now helper T cell pun jalan. Oh, you tengok, oh, dia ada antigen dekat sini. Okay, dia bind to me. Okay, so the antigen receptor of the helper T cell bind to the antigen that display by the macrophage, and then there are also the accessory protein CD4. Most important thing, you need to write CD4. CD4 accessory protein bind to the Cas2 MHC molecule. Then it bind it, okay, APC no, already know, okay, helper T cell already come, so it will release cytokine IL-1, interleukin 1. The full name is interleukin 1. Short form is IL-1 lah. Release it, and then it will activate your helper T cell. So this is what we call as activation of helper T cell. Helper T cell already know, okay, I already know this is coronavirus. I need to send this information to my bodies. So you can imagine that macrophage is like the initial army. They just ronda. Dia macam polis lah, ronda sini, ronda sana. Oh, dia nampak macam, oh, ini pelik-pelik punya. Oh, dia tangkap dulu. Okay, tangkap dulu, bagi tahu helper T cell. Okay, helper T cell already tahu, ini musuh. Saya kena serang dia. So, helper T cell will send the signal to B cell and cytotoxic T cell. Bagi tahu body dia, kawan, ini coronavirus, ini musuh tau. Awak kena bunuh dia. Okay, so it will release the signal to B cell and cytotoxic T cell. So how they activate here? So it will activate by release cytokine IL-2, interleukin 2. So the activated helper T cell then will release cytokine interleukin 2 for proliferation. Of course, it need to proliferate, meaning that it need to reproduce more and more activated helper T cell. Not only one, right? But then dia akan produce lebih banyak activated helper T cell. So dia boleh bagi tahu banyak body dia. Because only one, you imagine only one helper T cell just only one can quickly call all the B cell and cytotoxic T cell to activate. Cannot. So it also require large amount of number. So So it is like the snow effect become more serious, more and more. The number of the activated helper T cell will be increased in large amount. So uh so 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 this will release secret interleukin 2 as well to the B cell and cytotoxic T cell for the immune response. So when for the B cell, when it activate, it will trigger humoral immune response. We call as humoral immunity. So B cell will produce antibody lah. Cytotoxic T cell is cell-mediated immune response, which will attack the infected cell. So there are two cells. So we will see one by one. So what is happening in cell-mediated immune response? Okay. So at the same time, your cytotoxic T cell, right, once your cytotoxic T cell already release the signal by the helper T cell interleukin 2, okay, that he already know coronavirus is bad. Need to kill the coronavirus. But cytotoxic T cell does not kill virus. It kill the infected cell. You need to know coronavirus also can infect cell, right? So normally in coronavirus, it infect our lung cell lah. Lung cell, right? No, majority is lung cell. So once the coronavirus infect your lung cell, infected cell, it will also display the antigen fragment. Similar like the macrophage, but the antigen fragment replaced is by class 1 MHC molecule. Macrophage is class 2. Infected cell, all the healthy cell, infected cell display is class 1. So cytotoxic T cell containing antigen receptor will bind to the receptor, bind to the antigen that display by the infected cell, and also it containing the accessory protein CD8 bind to the class 1 MHC1 molecule. Once it already bind to it, it will vagonization occur. Then your cytotoxic T cell will starting to release the perforin enzyme. Perforin enzyme here like this. Starting release the perforin and destroy the infected cell. So it will why the perforin can be destroy the infected cell, because it will form the pores in the infected cell of the plasma membrane. So dia akan buat lubang lah. Dia akan buat lubang-lubang semua dekat dalam ni. So it is becoming hypotonic. So all the water from the outside diffuse into the cell. So your cell is hypotonic now. Then of course it will resulting in lysis process lah. Because if your animal cell immersed in the hypotonic condition, meaning that it will lyse. Right? It will become denatured. It will destroy the cell. So the function of the cytotoxic T cell is to destroy the infected cell. It does not destroy the pathogen. Dia tak akan bunuh virus. Dia bunuh infected cell. So this is a cell-mediated immune response. Okay. So another one is humoral immune response. So it will produce antibody. Okay. So what is happening here? Okay. So so this is the first step, right? Helper T cell, antigen presenting cell. After that, for B cell. So you need to know that it is highly specific. So B cell also require to bind to the same receptor. Meaning that the B cell that not yet activate and also not able bind to the alpha T cell yet, the B cell alone also require bind to the same antigen first, same virus. Like coronavirus, the B cell receptor also need to bind to the coronavirus first, and then it will display the like the macrophage. You can imagine that the initial step is like the macrophage. It will bind to the same pathogen. Most important thing is you need to use same. Don't just say pathogen. You need to write the same pathogen like helper T cell as helper T cell, because it is highly specific. So it bind to the coronavirus, and then it will lyse the coronavirus and display the antigen of the coronavirus. Once it display the antigen of the coronavirus, activated helper T cell also helper T cell will bind to the MHC2 antigen complex of the B cell. So B cell like the macrophage. We can imagine the similar function. It also display the Cas2 MHC molecule like this for the B cell, and then the activated helper T cell bind to it. Bind to it. Bind to it. So it was starting to activate the B cell. So it will also release the interleukin 2 lah. Similar interleukin 2 to the B cell. So the B cell starting to activate. So the activation of the B cell require 2 steps. The first step is B cell alone need to bind to the same pathogen virus, same pathogen like activated helper T cell one. Then it display the antigen MHC class 2 complex on the surface as the antigen presenting cell. Then the activated helper T cell bind to it and release the interleukin 2, activate the B cell. So the B cell then it will proliferate into the plasma cell. We call as plasma cell. Now it is not a B cell yet again, it becoming plasma cell, differentiate into plasma cell. Then this plasma cell was starting to produce antibodies. So this is the key point. Want to activate B cell require 2 signals. Once it already activate, it will produce plasma cell. And this plasma cell will secrete the antibody for humoral immune response. This is what we call as humoral immune response. Okay. So this is the overall structure. Once you already have the antigen taken by antigen presenting cell, helper T cell bind to it, and then it will release the signal to cytotoxic T cell and B cell. So once it release the signal to cytotoxic T cell, it will become activated, and then this cytotoxic T cell will lyse the infected cell. Okay. However, the most important thing is memory. In adaptive immune response, the special case is memory, meaning that they have memory. What does this mean? When you infected by coronavirus, okay, this first time you infected, so you already down the cytotoxic cell, B cell, okay. Now after that, after few weeks, okay, all the coronavirus already settled, finish. Okay, your body doesn't have any coronavirus anymore. Okay. If you exposed to the second time, meaning that you have infected coronavirus again, COVID again, second time. But second time, you will be more respond more faster. Why? Because of memory. Because in the first time, when your helper T cell be activated, it does beside it activate B cell and cytotoxic T cell, it will also produce memory T cell. So produce memory T cell. So this memory T cell already got the information about the coronavirus. So once you get the coronavirus, the helper T cell don't need to bind to the antigen presenting cell again. It don't need to wait until the antigen presenting cell, the formation of the antigen presenting cell to activate the adaptive immune response. It already have memory helper T cell. Or once it already received, okay, got the antigen again, any antigen coronavirus, then it will directly activate the memory B cell and memory cytotoxic T cell directly. Don't need to wait, because the adaptive immune response, why it take longer time? Because an antigen presenting cell. Because you need to wait the antigen presenting cell display the antigen, and then all that require time. At that time, your coronavirus already infect many cells already. So that's why that the person normally, if they get the second infection, they will recover more quickly, because your adaptive immune response is rapidly occur, because due to the formation of helper T cell. So in the examination, majority of the candidate not mention about the memory helper T cell. But you should mention. Not only helper T cell, B cell also, cytotoxic T cell also. You need to mention. Once they already activated, you not only just write plasma cell, you also need to write, they will also differentiate into memory B cell and memory B cell as well. And then in the final sentence of your examination, final sentence, especially in essay, you need to know the function of the memory B cell, cytotoxicity cell, and helper T cell. What are their function? The major function is that when they have the second exposure, second infection, the humoral immune response and the cell-mediated immune response will be rapidly occur because of the memory B cell. Then you just explain all that. Yeah. So you need to explain about memory. Because you will refer to the MPM report for this chapter. MPM normally will say majority of the candidate does not mention memory cell. So you should mention. Because MPM already state, you need mention. So you should mention. That's all about the development of the immunity. So this is very important in this chapter. And the majority of the question is coming out based on this chapter. Back on the topic. Another one is antigen antibody reaction. Actually, this is removed from the new syllabus, in the new syllabus, because old syllabus got mentioned, want to know, but in new syllabus they already removed it. But I think although it removed it, but I think you should know lah. You should know all because that one you already learn in SPM, neutralization, agglutination, precipitation. The new thing is here lah, activation of complement system. Although it removed, but I just include as well lah. Because affect maybe MPM coming out. Okay, coming out. So neutralization, agglutination, precipitation, I think I don't need to explain much lah. You already learned in SPM. I'm more focusing on complement fixation lah. Because complement fixation. So normally this one is the antibody work together with the protein of complement system, which we call as complement protein, like this. Once your antibody will bind to the antigen on the surface of the infected cell, it will activate your complement system. So meaning that you can see antibody bind to the antigen, so it will activate the complement system. So it will form the pores in the plasma membrane of the cell. Hence, water can go inside the cell. So the cell will short and light. So complement fixation is like the alternative way for the cell-mediated immune response. Look like cell-mediated immune response because it does not attack pathogen. Agglutination, precipitation, neutralization, they attack pathogen, they attack virus, they attack the pathogens one. But complement fixation is not attack the pathogen. It attack the infected cell. It make the infected cell die. Like this. So next we will go to the third topic for this chapter, concept self and non-self. Okay. So you need to know, as I mentioned during the development of the T cell and B cell, in our body there are antigen. Antigen is not only harmful, there are also the self-antigen, which we call as self. Our body also can display the self-antigen one. So you can imagine that in what you learn in the cell-mediated immune response here, in the real case, normal healthy person, not only infected cell will display the gas-1 MHC molecule with antigen. Normal cell also will display one. They also can display gas-1 MHC molecule with antigen. But this antigen is good. It's self-antigen. It is normal one. So, if the cytotoxicity cell is abnormal, unable to differentiate self and non-self, meaning that it bind to it, okay, it trigger the cell-mediated immune response, it kill this healthy cell. By right, it should not kill, but it kill. So meaning that this is the problem happen. So in our body system, it able to differentiate self and non-self. So our lymphocyte is able to detect any foreign substance which incompatible with it trigger to immune response like this. Our lymphocyte major rule. So the foreign substance is considered as non-self. Non-self. So if our any foreign substance is compatible to our body cell, then no immune response is triggered. This foreign substance is said to be a cell substance and is not rejected. Meaning that if the substance, the antigen is compatible to our body, does not causing any harm, this is what we call as cell. So the ability of the immune system to recognize self and non-self is known as self-tolerance. The terms are self-tolerance. So if the immune cell unable to recognize self and non-self, it will cause autoimmune disease, what we call as immune disorder. So establishing self-tolerance is very complicated. Why? Because self-tolerance is happening during the development of B cell and T cell. What I say in T cell is develop negative selection or positive selection, and then also B cell also got their self-tolerance. So so this is very important. So MHC play the role in recognizing self and non-self. Okay, so we know that in the development of immunity, you see, right, they have two class, MHC1 and MHC2. Okay, so MHC2 is very easy, because it only found in macrophage, B cell, and dendritic cell. Only these three cells display MHC class 2. Okay, so if you still remember, MHC is one type of the protein. It is one type of glycoprotein expressed on the cell, right? So in MHC class 2, only macrophage and B cell display that in your syllabus that you already know. Why? Why? So they display the MHC class 2 and antigen at here. So macrophage and B cell, they display the MHC class 2 for who to bind to it. Who bind MHC class 2 during the development of immunity? Who bind MHC class 2? The answer is helper T cell. Only helper T cell can bind to MHC class 2. Why? Because helper T cell containing CD4 accessory protein, CD4 receptor. So only the helper T cell can bind to MHC class 2. Only to activate the immune response. So it play the role. So the function of the MHC class 2 is play the role for antigen presenting cell in present the peptide to helper T cell. Present the antigen to the helper T cell. So it contribute the self-recognition by displacing processed antigen from engulfed material, helping helper T cell to recognize whether this come from the body or for an invader. So your helper T cell is able to differentiate. Differentiate. So when your macrophage B cell display the antigen, okay, it displays antigen here. So this antigen can be cell or non-cell. You don't know. You don't know what. So the helper T cell able to recognize it, helping to recognize it. So this is what you call as MHC class 2. Another one, MHC class 1. So MHC class 1, when during the development of immunity, what you see, when you see it, only when infected cell. During infected cell, you see the MHC class 1, right? During the cell-mediated immune response, you use MHC class 1. So MHC class 1 is all the nucleated body cell will display MHC class 1. All the cell in the human that containing nucleus will express as MHC class 1. So you can see majority lah. Majority all the cell will display MHC class 1. Class 1. So it play the role for the cytotoxicity cell recognition. So when cytotoxicity cell, which is a T8 cell, recognize it, when it recognize it as non-cell, meaning that or it will starting to trigger the cell-mediated immune response. So MHC class 1 play major role in cell-mediated immune response. MHC class 2 play major role in activating adaptive immune response. Class 2 is the initial one. Class 1 only for cell-mediated immune response. So this is the key terms that you should know it. You should know it. So that's why that MHC play the role in recognizing self and non-self. Okay. Now you need to know why. What T cell already go find at here, and then it just recognize MAC already know, oh, okay, this one is cell, and then each one is non-cell. How they recognize it? Okay, so we will go through more detail that because of the T cell development, development program to recognize peptide only when they are presented by the individual own self-MHC molecule, a property known as the self-MHC retraction. Because MHC, different individual have different type of MHC. The gene, the sequence, the protein sequence of the MHC, different type, different people have different MHC one. Like me and you, I have A MHC, you will have B MHC. Our protein sequence are different one. So when your MHC is founding to at my body, the helper T cell will recognize the MHC as non-cell. So although it is same MHC, we so-called MHC, but the sequence is not same. It's not safe. So that's why they want to relate the concept cell and non-cell to the organ transplant. Later we will see about the organ transplant one. So this is the explanation organ transplant. Okay. So during the positive selection, during the development of the T cell, your mature T cell is able to differentiate self-MHC and non and self-MHC, and many of these will have high affinity for self-MHC displaying for N peptide, for N antigen. Meaning that in the positive selection during the T cell development, your T cell is already be trained that, oh, they already know this is your one, only yours. Apart from that, they recognize as non-self. Only this we recognize as self. So when they have other type of MHC present in your body, in my body, containing the peptide derived from the allogenic cell, it may look like MHC, but this MHC is not yours because the protein sequence is already different. So therefore, recognition of other MHC is the example of the immunogenic cross reaction. So the process of the negative selection in the thymus will eliminate cells that strongly recognize self-MHC. MHC. But there is no opportunity to selectively eliminate T cell that have high affinity for allogenic MHC molecule. Meaning that when they have any outside sources of the MHC coming into your body, your T cell cannot recognize it. Because during the negative selection, they already eliminate it. Eliminate it. Because in negative selection, in real situation, your MHC, your helper T cell, will not bind strongly to the MHC class 2 one. Because you will still remember that, if you want to trigger the immune response, your helper T cell not only bind to antigen, they also bind to the accessory protein. If this accessory protein cannot bind to this class 2, then no immune response. So in the healthy situation, this macrophage display the self antigen, your antigen will not bind to it. bind to it. So it will not bind to it. So no immune response here. But if this is the non-self antigen here, non-self antigen receptor will bind to it. And then of course your CD4 also will bind. So this CD4 will bind to this class 2 MHC molecule. So this is the what overall situation. But for now here in the situation that if there are other type of MHC class 2 happen founding into cell, meaning that they would bind it more strongly compared to other type. When it bind more strongly, meaning that it will trigger adaptive immune response, even though the antigen is considered as self or non-self. So this is very important. So many clones of the T cell are specific for different for M peptide bound to the same MHC may cross-react with any one allogenic MHC molecule as long as the allogenic MHC molecule resemble compact of self MHC plus for M peptide. So that's why. So as a result, many self-MHC restricted T cell specific for different peptide may recognize any one allogenic MHC molecule. So meaning that many self-restricted T cell, MHC restricted T cell, meaning that the T cell cannot recognize this type of the MHC already. So it will display many different type of the pathogen may recognize any one allogenic MHC molecule. So this is what we call as. By contrast, in the case of the infected cell, only a small fraction of the cell MHC molecule on the cell surface will carry a 4N microbial peptide recognized by the host T cell. These are the main reason why allogenic cell evoke strong T cell reaction. So this meaning that if there are the outside sources of the MHC present inside in your body, it will trigger more serious immune response compared to the self one. So that's why MHC play the major role in recognizing self and non-self. Because in healthy, like the normal one, like us, normal one, MHC class 2 only can be displayed, recognized by the helper T cell, whether this comes from the body or foreign, because your helper T cell is able to differentiate it. So when it is coming from the inside one or your body one, then it will not trigger adaptive immune response. But when this MHC class 2 is coming from any don't know siapa tak tahu siapa punya. So why this happen is organ transplant. Because during organ transplant, your cell MHC and the donor MHC is different. So that's why it will cause rejection. So let us go through the organ transplant. So organ transplant meaning that there are 4 major type of auto transplant. Autograft is they will not have any rejection because it's from your body one, because it just transfer the tissue from one side to another side in the same individual, maybe from your hands from your leg skin to your hand skin, like that. Normally it's skin skin transfer. Isograph is for identical twins, like the twins are identical twins. Allograft is the most commonly found in the world. Normally, organ transplant normally is allograft, meaning that it is from the different member but in the same species like human. Like I want transfer the organ to you. I want to donate my kidney to you. In the same species. Xenograft is it only happen in the research stage only, still not yet in the market yet, normally in research stage, meaning using xenograph in different species. Normally they use animal, right? We will saw some news. They use the pig. What right in Europe country or US, if not mistaken, they transplant the heart from the peak to the heart attack individual and successfully already. The one of the news lah. Xenograft normally is in research state, still not approved yet by the medical association in the world lah. Normally because this were causing many ethical issue, yeah. But for now is allograft. So we were more focusing on allograft. So what is happening in rejection? So normally rejection is happening in allograft. Xenograft, of course, got rejection, but we were focusing on allograft. So this is a rejection process. Meaning that if the individual, the recipient, is accepted to that donor organ, meaning that there are no rejection one, just like the normal one, or the blood capillary is formed like the normal one. But when there are the rejection here, you see that in that the grafted tissue happening at here, and then it will cause many inflammation process, and then causing more worse. More worse. We will thrombosis and necrosis will happen. Meaning that we will make more serious lah. More serious lah. Actually, we want to receive an organ, it should become more healthier. But for now, become more worse. So you need to know that tissue that shares sufficient antigenic similarity, allowing the transfer without immunologic rejection, are said to be histocompatible. Meaning that there are no rejection at all lah. We call it compatible. Normally this happen in identical twins. If you want to have 100% no rejection, identical twin only. Identical twin or autograph only. Only these two. However, tissue that display significant antigenic difference are histoincompatible and induce immune response, lead to tissue rejection. Then this is allograft. Okay, so what is happening in allograft rejection? So you can see this diagram. When the donor organ, like if a donor organ, the donor me, if I want to donate the organ to you, like donate the kidney lah, I want to donate the organ and transplanted into the recipient, you can imagine that this kidney is already from the B base, like A and B lah. A is recipient, B is donor. This is the B kidney in the A body. So your MHC sequence is different at all. Your MHC is different. So this will cause the recipient immune system recognize this organ as non-self, triggering the adaptive immune response. That's why I say that MHC is play the role in recognizing self and non-self. Because your MHC sequence is already different. Although it still look like MHC, but the sequence is different. Your body, your helper T cell is unable to recognize this MHC as self, although it looks same. But after they bind to it, imagine it bind to it this class 2. Because you need to know that macrophage, you can imagine in the kidney, in your kidney part, macrophage, okay, dia jalan sini, dia engulf the antigen, engulf the NET and display the antigen. And maybe this antigen is from the kidney one, from the A person. From the donor kidney, donor's kidney. So meaning that this antigen is already considered as non-self already. Right? Because this antigen is already non-self. Or they display the self antigen. Okay, this antigen is self one, but your T cell receptor still can bind it. They just bind to it. Okay, bind to it. If this is non-self, it is self and bind to it. Once it bind to it, your CD4 will bind to it. So it will recognize. Your T cell receptor will recognize first. Is it self or non-self? Okay, if it is non-self, it will bind tightly. If it is self, then your antigen receptor will stop it lah. But if it is non-self, your antigen receptor of your helper T cell bind to it. Okay, now CD4 accessory protein starting to bind, and then it will recognize it as non-self also. So once it already recognize as non-self, then GG, all this happen. So you can imagine that, in the activation of the T cell, generation of the T cell by direct, indirect, and indirect antigen presentation, transport to the allo antigen to the lymph node, and all the rejection happening at here. So allograft rejection containing two pathway, either is direct pathway or indirect pathway. Okay, so what is happening is sorry direct pathway. So there are two pathway of the presenting allo antigen on the transplanted donor organ to the recipient T lymphocyte. Okay, so either direct or indirect. So direct meaning that this is your allogenic MHC, meaning that this is from the donor one. So your T cell recognize unprocessed allogenic MHC molecule on graft APC. APC. So meaning that your transplanted organ containing donor derived APC, APC. So which play a role in initiating in this immune response. Okay. So like this, you can see this diagram. When you are this kidney, they're containing the donor kidney itself containing APC. They containing their own macrophage. Easy to say it. It containing the macrophage. So this macrophage is not your macrophage. Can you know get right? In your body, you have your own macrophage, right? You have your own macrophage. Okay. For now, you receive the kidney, and the kidney also carry their own macrophage. That macrophage is donor one. Your donor macrophage. So the donor macrophage now enter into the kidney, into your body. So this macrophage, they still function. They still function. Or they will go to the lymph node, go to the donor, your lymph node, donor lymph node, go to there. Migrate to the lymph node by blood where they activate the effector T cell. Meaning that this macrophage still can function as antigen presenting cell. Right? You can still function as antigen presenting cell. So it is already considered as non-self already. So it will cause your recipient helper T cell can be activated by binding to antigen fragment complex with regards to MHC molecule. So MHC molecule. So it already uh, disrupted during the transplantation. This migration occur primarily through bloodstream rather than by lymphatic vessel. So once activated, your allo-reactive helper T cell will circulate back to the graft directly and attack the transplanted tissue by humoral immune response and cell-mediated immune response. So this is recalled as direct recognition. So this is where the kidney carry their own macrophage. So it containing the recipient macrophage. So this recipient macrophage can be activated by the donor macrophage. Donor macrophage. So you can imagine like see lah, allogenic APC. This is a allogenic APC. So it also display MHC. And then this is your own cytotoxicity cell. Cytotoxicity cell, it recognize it. Recognize this allogenic APC cell. Bukan Dah recognize macam, ni bukan awak. So dia akan activated cytotoxicity cell. And then at here, helper T cell. Your helper T cell also will recognize this. Pun nampak ini bukan ini is considered as non-self. So it directly activate. So ini pathway dia lah. So you can imagine that the donor, the recipient macrophage now is as pathogen, like a pathogen now. So it can be activated by your own human system. Your helper T cell and your cytotoxicity cell will be activated. So this is what we call as direct pathway. Another one is indirect pathway. So it does not contain any the recipient does not carry any dendritic cell, or the doctor is very excellent. They already all remove all the recipient dendritic cell, meaning recipient macrophage lah. All the macrophage, dendritic cell, B cell, all are already removed. Very good. Okay. Now the problem is they still got any allogenic protein. Your kidney also containing allogenic protein, which we call as antigen. Allogenic antigen, meaning that in the recipient body is normal one, but not recipient, donor this donor, they stay their body is normal one. The antigen present for them is normal, but for when it goes through the recipient body, meaning that this is the something problem. This is what we call the antigen is considered as non-self. So non-self. So the recipient own antigen presenting cell will recognize this peptide. So this peptide is considered as non-self. So once it recognize this considered as peptide as non-self, meaning that it will activate the helper T cell. So it will directly attack the organ. Also will trigger the adaptive immune response. So this is what we call as indirect. Because the activating of the immune response is due to the allogenic peptide. So this one is not considered as MHC lah. There are no other MHC come from, or no MHC, but the antigen is different. So in recognizing self and non-self, so you need to know in recognize self and non-self, there are two important factors. First one is antigen. Either this antigen is from your own body or from other body. If it is from your own body, there should no any recognition. Your B cell receptor, your helper T cell receptor will not recognize it. But when it is non-self, even though your MHC class 2 is self 1, they still can recognize. Right? Okay, that one is first part. Second part is MHC. MHC, this one is more focusing on organ transplant. Okay, because in the normal situation, all your MHC is similar. But when there are other MHC coming to your body, I'm focusing on MHC class 2, coming to your body, meaning that MHC class 2 is considered as non-self. So even though the peptide, whatever the peptide is self or non-self, it already can trigger the immune response. Because your helper T cell already tell you, this is not. Your CD4 accessory protein already tell you that it can bind to it. Already tell you it not. So it will cause more serious infection. So that is why that direct pathway will be more serious compared to indirect one. Because direct pathway is simultaneously pathway. It not only activate the helper T cell, it also activate the cytotoxic T cell, and it simultaneously activated. So that's why lah. So you can see here, direct recognition is the allogenic antigen presenting cell, and this is your allo-reactive T cell, meaning this is the donor recipient T cell bind to it and then causing directly bind. But indirect is using your own T cell. So so this is a direct and indirect lah. So you can see indirect like this. This is a recipient, right? Recipient macrophage, and then once it recognize the bind to the allogenic protein, display the allogenic antigen, it will activate the helper T cell, all that. Okay, so now how do we minimize the rejection? We know that rejection majority occur. So there are many drugs now. So this is a few example drug to reduce the rejection. So if you want to reduce the rejection, is to prevent the rejection through the immunosuppression. You need to suppress the immunology, inhibit the T cell activation and the adaptive immune response. If your helper T cell not activate, then no activate immune response. So this is a few drug example and their mechanism of action. I think this you can read by yourself. I don't need to explain much. Yeah. Okay. Next is the final one is the immune disorder, which is autoimmune disorder. So autoimmune disorder is defined as the inappropriate immune response to self-antigen that lead to tissue damage and disease. Meaning that your immune response is already failed. Even though it is self-antigen, but they still attack yourself. This is what we call as autoimmune disorder. Okay, autoimmune disorder. Okay. So there are actually two major type of the autoimmune disorder lah, organ specific or systemic. So in our syllabus, you will learn about type 1 diabetes mellitus, and then another one is multiple sclerosis, and then the final one is HIV infection. HIV infection. Infection. These three only. Multiple sclerosis, type 1 diabetes, and HIV. In old syllabus, there are also another one is SLE. Systemic lupus erythematosus. But this one is already removed in your old syllabus, in the new syllabus. But they add on two new disease, multiple sclerosis and type 1 diabetes mellitus. This one type 1 diabetes mellitus, as I mentioned, is actually continue topic from the last chapter, homeostasis. Homeostasis. So in homeostasis, I not mention much. Here I will explain more detail. Okay. So first of all is type 1 diabetes. Okay. So what is happening in type 1 diabetes? So this is a comparison between healthy and type 1 diabetes patient. So it is actually autoimmune disease which your antibodies destroy the beta cell. Destroy the beta cell in the pancreas here. Okay, so this is a healthy one, meaning that you produce the B cell, produce the B cell, and then B cell, and then your immature autoreactive B cell, and you will go through all the normal development, and then no rejection at all. But in the type 1 diabetes, it produces the autoreactive B cell. So this autoreactive means that this B cell is already can destroy the abnormal. We call as abnormal, autoreactive, abnormal already. So the scientific terms so this autoreactive B cell it cannot do all this tolerance, self tolerance all fail. They should do this all tolerance first, but this all tolerance fail. So what happened? It will directly destroy the beta cell. So your B cell can be destroyed, can secrete the antibody to destroy the beta cell in the pancreas. So your T cell able to destroy the beta cell, resulting in insulin deficiency. So efficiency. So this one is another diagram lah. So your islet or Langerhans cell containing several cell type, right? Alpha, and beta. Alpha is for glucagon, beta is for insulin. But in type 1 diabetes, your effector T cell recognize the peptide from a beta cell is non-self. So it trigger the immune response so that it destroy the beta cell, so no insulin can be produced. No insulin can be produced. So the process begins when your antigen presenting cell, like the macrophage, in the pancreatic lymph node within the or Langerhans cell, and then presenting the beta cell, self antigen and secreting the inflammatory mediator. So this will trigger the adaptive immune response, which activate your autoreactive helper T cell. helper T cell. So your B cell play a critical pathogenic role, not primarily through antibody secretion, but efficiently capturing islet antigen site insulin and presenting them to autoreactive T cell, T cell, so that it can cause the T cell to destroy all the beta cell. So this is happening lah. So you can imagine that your helper T cell, your B cell, all are already abnormal already. So we call as autoreactive. So it will directly kill your beta cell. So if your beta cell is already killed, no insulin lah. So this is what we call as type 1 diabetes. Type 1 diabetes. Okay. So yeah. So this is more complicated one lah. So I just show you lah. Right? So this is bone marrow. It will produce the cell. B cell and T cell, right? B cell and T cell, and then thymus develop the T cell, and then after that it will produce central tolerance. After that it will go through the peripheral blood, and then it will directly kill the macrophage. Macrophage, right? This macrophage will will bind to the display the beta cell antigen, causing the helper T cell be activated, and then causing the beta cell to kill all that. Yeah. So this is more complicated one, but I think this one I just share it to you for the more detailed explanation. So actually you don't need to know all this detail. You just want to know that like this only, just enough. You just want to know that your helper T cell, your B cell, your T8 cell, cytotoxic cell, all already are autoreactive. So that your helper T cell bind to the beta cell antigen, and then it will activate your B cell again, and then this activate B cell will secrete the antibody, destroy the beta cell, and also destroy the beta cell, and then your CD8 T cell also will destroy the beta cell. So all this effect is to destroy the beta cell. So this is type 1 diabetes mellitus are causes of the type 1 diabetes mellitus. So the treatment including injection of insulin. So the primary goal in insulin therapy is intensive to control the blood glucose level at close to normal while avoiding hypoglycemia. So your insulin is injected normally is by string or insulin pen called subcutaneous injection. So there are two major types of insulin which we call as basal insulin and paranal insulin. So basal insulin normally is to provide a steady level of the insulin to manage the glucose between meals and overnight, which is the during period of fasting. So normally is to maintain your insulin level overnight, because for the patient that does not have insulin, meaning that when your blood glucose level is highly increased, right, and no insulin happen, you will get the hypoglycemia one. So they want to avoid hypoglycemia. So this basal insulin is to maintain the long period. So it provides a continuous low level of insulin to keep your blood sugar level stable when you are not eating. So it is more sustain more longer lah. But for the prandial insulin, normally is used to manage rapid change of your blood glucose level when you eat. So when you eat, your blood glucose level will rise very fast, right? Rise very fast. So that they use insulin to reduce your blood glucose level. But now you don't have insulin. So you use this prandial insulin so to control the glucose spike causes by the ingestion of the food. Digestion of the food. So this insulin is provide the shorter period lah, because it is not just not to control the blood glucose level for a long time one. Just want to control the blood glucose level when you are eating only. So this is what we call as prandial insulin. Okay, so beside the insulin level, another more advanced one is beta cell replacement therapy, which that they replace the beta cell. So the therapy considers specific patient with type 1 diabetes to restore the body natural ability to produce insulin, meaning that they replace the beta cell. Your beta cell now is already destroyed, so they replace another new beta cell to you. So lead to the occultation of the insulin therapy and attainment of the normal blood sugar level. So either they transplant the pancreas or they transplant the islet cell, your beta cell lah, meaning so these two lah. Okay, so yeah, so that's all about the type 1 diabetes. So then another one is multiple sclerosis. Okay, so it is actually the T cell mediated disease, neurologic disease caused by a destructive immune response against the central nervous system myelin antigen, including myelin basic protein, proteolytic protein, and myelin oligonucleotide glycoprotein. So due to these three protein, meaning that these three protein actually should be normal. This protein is normal one, what we call as antigen lah, normal antigen. But multiple sclerosis happen that attack these three protein. So it will damage along the nerve myelin sheath of the nerve fiber in the brain and spinal cord. So it is actually in your brain. It attack your brain. Your immune system attack your brain. Okay, so so we know that in myelin, we know that it function to insulate the nerve fiber, right? Myelin sheath. Myelin sheath insulate the muscle fiber to speed up your nervous system, generating the nerve impulse, electrical impulse. So when you have this kind of disease, it will break down the myelin sheath, lead to numerous neurologic dysfunction, ranging from numbness in the limb of the paralysis or loss of the vision. So it attack your myelin sheath. Here, myelin sheath. So in human, our neuron normally contain myelin sheath, but this disease destroy myelin sheath. They destroy the myelin sheath. So make your neuron does not contain any myelin sheath anymore. So when you do not have myelin sheath, what is the effect? Effect is you want to transfer the electrical impulse from here to here require long time. You need to know our nerve impulse, because human is very large, considered as large mammalian. So we need the myelin sheath to increase the speed of the electrical transmission of the nerve electrical transmission. But if no myelin sheath, it will cause paralysis problem. So the exact cause, actually the exact what caused the cell attack your immune system, attack the myelin sheath, actually is so far still not yet know. So far, in the real scientific way, still not yet. They just only know your T cell, autoreactive T cell, can attack the your nerve cell. So the exact cause is unknown, but most majority scientists agree that several triggers are involved, including genetic, gender, and environment. And so normally this disease occur in women more rapidly compared to men, and frequently develop in the childhood. For female, childhood normally they will occur at here. So if you want to as I mentioned, because for now there are no scientific yet why the multiple sclerosis happen. They only know that there are any unknown trigger the inflammation in your brain. There are inflammation happening in your brain, meaning that there are many white blood cell trigger not triggering gathering at here gathering at here. So this cause the blood-brain barrier become locally permeable to allow your autoreactive T cell migrate into the brain tissue. So because of the inflammation happening in your brain, it allow your autoreactive T cell enter into your brain tissue now. So this autoreactive T cell will encounter their specific alter antigen receptor displayed on the MHC2, like by bacillus microgalia or infecting microphage. So this reactivation causes the T cell to release the cytokine to activate the adaptive immune response as well. So your inflammatory reaction occur in the brain due to mast cell activation, complement activation, antibody, and cytokine. So it will cause demyelination of your neuron occur. So this is actually what we say as the multiple sclerosis mechanism lah. And so first of all, it trigger the inflammation in your brain. Your brain suddenly trigger inflammation. So if there are inflammation happen, all the white blood cell, including the autoreactive T cell, gathering at there. Gather at here. So your macrophage will engulf all the antigen at here. So the antigen including here, MBP, PLP, or MOG. And then this autoreactive T cell, will bind to the cell, the nerve cell lah. One of the nerve cell. This in this example they use microglia cell. So in examination you can just use nerve cell lah. Enough. Bind to the nerve cell and then activate the immune response. So once they activate immune response, they will destroy the myelin sheath. So it will attack the myelin sheath, remove the myelin sheath. So your neuron now become unmyelinated. So the effect will be cause the paralysis of the limb and loss of the vision. So this is the whole process. So you can just write all the process like the adaptive immune response. Yeah. Okay. So this is the symptom. So this is the effect. For the patient that get this kind of disease, so this is a bad effect. So you can read yourself. You just memorize. Few symptom, then you just know lah. When the exam come out, especially objective question, when the exam come out, what are the symptoms that have in the multiple sclerosis? You should able to answer it lah. Okay, the final one, HIV infection. Okay, so HIV infection, although it is caused, also is one type of the autoimmune disease, but it is not caused by the autoreactive cell. The first two one, type 1 diabetes and the multiple sclerosis, both of them caused is by the autoreactive cell. Your lymphocyte is already abnormal. But now here is caused by virus, right? HIV virus. So HIV in the full terms is the human immunodeficiency virus. So you already know that human immunodeficiency. So immunodeficiency meaning that this virus can cause your immune system become deficiency. It will break down, destroy your immune system. This is why HIV virus is very dangerous. And also that is why that we call as HIV the person that get the HIV is not death, is not caused by HIV. It's caused by weak immune response. So HIV is the vector. So the person that get the HIV is recalled as AIDS lah, as acquired immunodeficiency syndrome. So yeah, of course you need to know what is HIV. So HIV is an example of the retrovirus. Retrovirus. So retrovirus is the envelope containing single-stranded RNA virus. So in virology, for those who are interested to study about virology, virus actually is very interesting organism. I cannot say it's organism, but it is interesting fact in the world lah. So there are many classes of the virus, including adenovirus, and then also there are also retrovirus, and then there are also other type of virus, Okay, so that one is later if you for you uh more easy want to know about the virus lah, when you want to study about the virology. So there are many type of virus including helical virus, polyvalent virus, spherical virus. In virus, so normally virus look like this lah. So in this one is I want you to know the major things that you need to know is that it is only have single-stranded RNA virus. The gene. The gene only have single-stranded RNA virus. So the inner part lah, you can see this is a gene, RNA virus. Okay, and it is envelope virus. Envelope virus. There are two type of virus. Either is envelope or naked. So this one, HIV virus is envelope virus, meaning that they containing the protein that capture surrounding of the virus. So they're containing the glycoprotein one. So there are two major type of the glycoprotein with GP120 and GP41. So GP120 is the outer surface one, like you can see like the moon shade one, and GP41 is at that attached to the envelope one. So normally GP1220 120 is external protein that play the role in binding to CD4 receptor. That one you will learn later. I will explain more detail during the HIV mechanism. Okay. Okay, so HIV contain the spatial enzyme we call a reverse transcriptase that transcribe RNA into DNA. This one is spatial to the HIV virus. We all not HIV virus. In human, we know that in DNA gene expression, we transcribe the DNA into RNA, right? And then later to protein. So in human, we does not have this kind of enzyme one. Reverse transcriptase is not found in human. We cannot use RNA to do DNA one. We cannot reverse the process one. But this virus enable them to do this. So this RNA DNA information flow that is opposite of the usual direction lah. Direction lah. So next we go through the mechanism. Okay. So how the HIV infect our cell. So the target cell of a HIV is infect helper T cell. It will not infect other type of cell. It just infect helper T cell. So HIV is very clever virus. We already know that a human immune system is controlled by helper T cell, because you need to know that if you want to activate adaptive immune response, you require helper T cell. So when your helper T cell is already infected, how they fight HIV? Cannot. Because if your helper T cell already infected, meaning that your B cell and cytotoxic T cell does not receive any information, then nobody can kill the HIV anymore. So this is a clever part of the HIV. So your GP120, the external glycoprotein, will bind to the CD4 protein of the infected of the helper T cell. So if you see the CD4, of course only helper T cell. Only helper T cell containing CD4 protein. So once they already bind to the CD4, your GP41 will bind to the chemokine receptor on the helper T cell, and fuse to the and starting to fuse into the helper T cell. So it was starting to release the capsid, which is what we call as capsid. So your capsid containing the single-stranded RNA virus and also the reverse transcriptase. So it will release the single-stranded RNA and also the reverse transcriptase. Okay, after that, it will do the reverse transcriptase, will catalyze the reverse transcription of the single-stranded DNA to form RNA-DNA hybrid. Meaning that it will form the single-stranded DNA first. Single-stranded DNA. After that, your RNA template is partially degraded by the ribonuclease enzyme, followed by the synthesis of the second DNA centrose to strand to yield HIV double-stranded DNA. So in simple word, after the single-stranded RNA and reverse transcriptase already released by the capsid, this reverse transcriptase will firstly synthesize single-stranded DNA first. After that, it was using the same enzyme to synthesize double-stranded HIV DNA virus. Okay, so this double-stranded DNA virus, double-stranded DNA HIV virus, HIV was diffused into the nucleus now, translocated into the nucleus, and integrate into the host chromosome by integrase enzyme, meaning that they are campur dekat sini. Meaning that this is a normal virus, right? Normal gene, normal chromosome, and it will integrate into it. So this red color is actually becoming HIV now, becoming a part of the chromosome now, like the part of the chromosome now. So this process is we call as integration by integrase enzyme. After that, it will do the all the like the normal situation like the normal one transcription, translation, using the host and host factor and using the transcription, perform the transcription and translation, produce the produce the mRNA. So this mRNA is not a normal mRNA, already become the viral mRNA. So this viral mRNA go to the cytoplasm and then do the translation process. So it will produce the viral protein. So the viral protein including the glycoprotein glycoprotein the GP120 and GP41. So it will the glycoprotein will assemble together into the membrane into the cell membrane to produce the new HIV virus. And at the same time also DNA replication also happen, right? DNA replication also happen to produce the produce the new RNA virus. RNA virus. So this RNA virus will assemble together with the viral protein to produce new HIV. So the membrane will bud out, forming the viral envelope. So the released viral particle is completely the maturation incorporated with the protein. And new virus is released, and this new virus will affect other type of the helper T cell. So this is a whole mechanism that happening in HIV by HIV that infect helper T cell. So how HIV cause AIDS? AIDS. So this is a diagram you can see. So what does this diagram be? The blue color one is your HIV concentration, your virus concentration. Red color one is helper T cell concentration. And then the red color one is anti-HIV antibody concentration. Okay, so you can imagine that when you get the HIV or you get the HIV, you suddenly increase. After that, it decrease back. Why? Because your helper T cell play the role now. Play the role now. Not only helper T cell, but your secondary immune response. Your macrophage, your neutrophil, your dendritic cell will play the role to eliminate the HIV. But at the same time, your helper T cell also increase. But when your helper T cell increase, it will cause HIV infect more and more helper T cell. So you can see that at the end, where the year and year, your HIV increase and increase. Finally, when it increase until certain amount, until your helper T cell already cannot be produced anymore, you will cause AIDS. So when you get the HIV, you will have the latent period. So normally your latent period is almost 10 years. Can be 10 years. So during this 10 years, you may not have any symptom, but when you have a symptom, you already very serious, I can say. And finally, if you get a serious AIDS, if you not get any treatment very well, finally it's death. And the death is not due to HIV, the death is due to weak immune response. Because your helper T cell is too low, meaning that your other type of the pathogen, other type of virus and bacteria infect to your cell, easily you can get severe inflammation and infection. So that's why that HIV is very dangerous disease in the world for now. So that's all for this chapter and also the topic for this chapter. Hope this video can provide a clear explanation video that you understand about the human immunity system in STPM Biology Chapter 13 Immunity lah. Okay, so if you have any question, just type in the chat box or join my Telegram group for the deeper discussion for this chapter. And thanks for those who are watching from the beginning until the end. And thanks for your supporting as well. So this will be the last two video for this semester. Next video will be the last chapter, chapter 14, infection disease. So that's all for this video and thanks. Bye.